And so hopefully you have now mastered mitosis and meiosis. And now we're going to go into some things that we can determine problems that might have happened with that and some of the disorders we see with that. So the first thing that we're going to talk about is a karyotype. Now, karyotype is where they take a picture of your chromosomes and they pair them up, and then they determine if there's any chromosomal issue. Are you missing a chromosome? Do you have an extra chromosome? Are you missing part of a chromosome? We cannot tell if you have a bad gene or not, but we can tell if you have a bad portion or segment of your chromosomes. So this is what it looks like. So we can see here that they've paired them all up nicely. They band according to, they stain them with different dyes, and so we can get those nice little banding patterns, so we can find the chromosomes, the homologous chromosomes that pair up. And then we can see that this person has 2 chromosomes at every site. Looks good. And in fact, this would be a male. It has an X and a Y. Notice down there at chromosome pair 23, we can see that. So when we do a picture, probably the most, I don't want to say famous, but the disorder you're probably most familiar with is Down syndrome. It is called trisomy 21 because at the 21st position, there are 3 of those chromosomes. Now, we can all pretty much, when we see a child that has Down syndrome, it's very obvious, that type of disability that they have. Not only do they have the characteristic facial features, but they do have a low IQ. So they are short. They have other problems as well. They have heart problems. They have respiratory issues. Often they develop leukemia and have Alzheimer's. They do have a shortened lifespan. It is kind of crazy because the incident will increase with the age of the mother. So when we look at this next chart, I'm going to show you in a minute here. Here's the typical Down syndrome child here, and you can see there's 3 chromosomes here. But when you look at the age of the mom, and the problem with the mom is that when we are born, our eggs are already made. So they are made in utero while we're still in mom. And so those eggs they don't finish meiosis. They are paused in prophase 1, where they're hanging out with that homologous chromosome. And so the older you get, you can basically say the more expired your eggs are getting. And so they find it difficult to separate those homologous chromosomes, and you end up with an abnormal number of chromosomes in your eggs, and that is called non-disjunction, and that's what we're going to look here in a minute. And so notice how the older mom gets it's the numbers, they significantly go up. This one is really kind of scary because if you are, over 40, you're in the high risk group. You are a much greater chance of having a child with Down syndrome, which is probably why we're seeing a lot more children with Down syndrome as women are waiting to have children later in life. So non-disjunction is the failure of the chromosomes. It could be either the homologous chromosomes, we don't have a good divorce, they stay together, or the sisters do not separate. So we could have non-disjunction in meiosis 1 or meiosis 2. Either way, if that egg or sperm does not form properly, and that egg gets fertilized by either a healthy or a non-healthy or the wrong number sperm, you're going to have a child with Down syndrome, or that egg itself could be not properly made. So either case, you can have Down syndrome that is the father's fault too, but in most cases it is the mother. So here we have a picture of non-disjunction. So those homologous chromosomes did not separate, and now notice we're going to have 3 chromosomes going to the left and only 1 to the right. And so now we're going to split the sisters up like we normally would. And now we have these 2 gametes here have too many chromosomes, and these 2 gametes here have not enough. So here we have our divorce happens just fine, but the sisters failed to separate. And again, we're going to have a gamete with too many, one with not enough, but actually this side, we get some that are normal. So when we have non-disjunction and meiosis two, we have a 50% chance of a normal outcome and 50% chance of abnormal. And again, this is not all your sperm. It would just be like one sperm that, when it's being developed, one of what we call the basically the stem cells that will make sperm has that error in its process. Now, a lot of the times, most of the zygotes, the fertilized eggs that have a non-disjunction issue, will result in miscarriage. So most of the situations where you're missing or have too many chromosomes, they're not viable. They die. This is probably one of the big reasons why when you hear women miscarry was most likely because of a non-disjunction issue. But we have some that are viable, for instance, Down syndrome. Some other ones are some of the sex abnormalities. And so we're going to talk about XXY, which is Klinefelter's, XYY, which is also called Jacob syndrome. And then we have, you could say the super female here, or the X0 means it's missing. So all these have too many. In this case, they're missing a chromosome. That's called Turner syndrome. So I'm just going to kind of go through these. So Klinefelter's is where a man has two X's and a Y. So they have 47 chromosomes. They tend to have very little hair, facial or pubic hair. Their testosterone production goes down. They may develop breast. They have wider hips. They're very tall. They have abnormal body proportions. So here's kind of a picture of that. And so you can see here we have an extra X chromosome. This disorder is probably the best where you could say would be like a hermaphrodite. Each individual case is a little different as to whether they have more male traits that have developed or female traits that have developed. So a lot of times, again, with cosmetic surgery and whatnot, they used to just change them to the one that would have been easier to do, but with now modern medicine, a lot of times they'll wait for that individual to grow up and maybe make that decision if they want to be cosmetically male or female. Turner syndrome, notice here we're missing that chromosome. Lots of different characteristics here. Actually, most of the babies that have this particular genetic disorder do not survive. They are most of the time they are miscarriaged. Very few of them survive, and they usually do not live very long, in that usually young children. We also have what's called Jacob syndrome, which is you have two Y chromosomes. And with this particular disorder, we see learning problems in these males, delayed emotional maturity. We also see that they're tall and thin. They have acne and speech problems and reading problems, again, tied to those learning disabilities. So here is a karyotype of a Jacob syndrome. So notice here we have the X with the two little Y's. It was kind of interesting because it was originally thought that if you had Jacob syndrome, you had that extra Y chromosome, so you had more testosterone that led to aggressive behavior and the likelihood that you would be a serial killer. If you look at the number of serial killers we actually have in jail and sample those that had Jacob syndrome, even if they had one or two, we probably don't have more than 10 people in there. So the sample size was really small. It was really bad science. So if you hear that Jacob syndrome means that you have a higher chance of being a serial killer, no, that study was totally thrown out of the water. If you were to look at the number of Jacob syndrome individuals that are not in jail, it would totally speak otherwise. So this is again where you see things on Facebook or other types of media that are wrong and they get refuted, and you never see that, and they'll pop back up and you'll get that bad information again, but the refuted part does not show up again. So just know this was an example of bad science. So with evolution, we have these errors in mitosis and meiosis that can lead to what we call polyploidy species. And that's where they have more than two chromosome sets. Now, in animals, this is generally not viable, but in plants, it gives us like these super sized leaves, flowers, and fruits. So here would be actually a human that is tetraploidy. Notice they have three chromosomes that triploidy, not four. You can have tetraploidy. You do in plants. And so I think we Googled it at one time, and I think they'll, again, these are all non-viable. I think we found that the oldest a human triploidy individual lived was 10 days. So generally, again, these are all miscarried or they don't live long. We also have things that can cause chromosomes can break. They can sometimes reattach to other chromosomes. Some of that information, as long as it's there, the cell is good. They may have problems with reproducing. They also those genes could be broken in locations that can cause problems, like cancer and such. So again, we can kind of look for those gross chromosomal abnormalities on the karyotype. Some other things that we can find sometimes is what's called deletion, a duplication, inversion, and translocation. Now, again, these are chromosomal abnormalities. We're not talking about genes. We're talking about whole segments of chromosomes being deleted or copied or flipped or moved to a whole different chromosome. We know that, like for hemoglobin, we have many, like over 20 copies of hemoglobin in our genome. This allows some of those genes to be tinkered with while still leaving us a good copy to make that very important protein that allows us to carry oxygen around our body. And where did okay, we're going to be goofy again. All right, so here is just a little picture. Notice here's a deletion. Again, most likely these are from uneven or a problem with crossing over. So we have the duplication here where both copies went to one chromosome. Here, again, it left that chromosome. In inversions where they got flipped. And a translocation is where if this is not a crossing over with the homologous chromosomes, this is basically you had a cheating pair of chromosomes. So they found a different chromosome, not their partner, and they swapped. And that again can cause problems. And then we have a little video here that I don't think is working anymore. And then we are done with chapter 8. All right.